Hi everyone. Dear students, in my last lecture, I have explained to you about the gametogenesis, formation of the male gamete and female gamete. Today, let's know about fertilization and implantation. Now, what is fertilization? Fertilization is a process during which the sperm and egg meet or fuse to produce zygote. Now, how does it happens? So actually during the copulation is nothing but the sexual intercourse. During that sexual intercourse or copulation, the sperms are released by the penis into the vagina of the woman near the cervix. So these sperms, when they are released near the cervix, they are motile, they start traveling upward through the cervix, uterus, and ultimately enter into the fallopian tube. Now, while they pass through the cervix and the uterus, especially when they are crossing the uterus, large number of sperms are actually destroyed or engulfed, eaten up by the phagocytes released from the wall of the uterus. So only few will actually be successful in reaching the fallopian tube. The sperms deposited in the female genital tract at one time, that is during the copulation, they are about nearly 200 to 300 million. But only a few thousand may be successfully reaching the fallopian tube. Okay, and one more thing happens, that as the sperms pass through the uterus, some chemicals released from the wall of the uterus will bring about the capacitation of the sperms. What is capacitation? Capacitation is the process, or it is the maturation of the sperm, in which from the plasma membrane of the sperm some of the proteins are removed so that the plasma membrane becomes more thinner and perfect for the fertilization, or easily it can fuse with the egg membrane. So that's why it is called as maturation of the sperm that takes place in the uterus. It's called as capacitation. Okay. Then the secondary oocyte, which is released by the ovary, that is picked up by the fimbriae and transported into the fallopian tube. Okay, and how this happens? It is by inward beating of the cilia. The cilia which are there along the fimbriae, just recall the female reproductive system, fimbriae, infundibulum, and then the fallopian tube. So these cilia will beat and create a kind of current which makes the egg enter into the fallopian tube, and inside the fallopian tube, the lining of the fallopian tube also has cilia, and that keep the egg slowly moving. Okay, now let us look into the diagram to further understand what we have just learned. Now look at this diagram. So the egg is released here. This A is representing the released secondary oocyte or egg, and these are the fimbriae by cilia on their inner surface. They create a current towards inner side so that the egg enters into the fallopian tube and it moves along the fallopian tube, because the lining of the fallopian tube also has got cilia. Okay, now this is the spot of fertilization, where you see that the oocyte or egg is surrounded by sperms. Okay, so in the ampulla region, this is what called as ampulla region. In ampulla region of the fallopian tube, actually the fertilization takes place. So let us go through the points further. An egg can survive only from 24 to 28 hours inside the fallopian tube. Okay, that is the time when, if the sperm enters, that will be fertilizing the egg in the ampulla region. In the ampulla region. Okay. Now fertilization takes place in different steps. Let us see what these are. The first one, we take it up here as the association of sperm and egg. So chemicals released from the egg called as the fertilizin, and that will be attracting the anti-fertilizin. That will be attracting the anti-fertilizin of the sperm. So this is the chemical attraction between sperm and egg, that is called as chemotaxy. According to the recent findings, though the movement of sperm from the place of deposition to the place of fertilization, it's around 10 to 15 centimeters distance, and this is covered by these tiny microscopic spermatozoa by the help of beating of the flagella. But how they could orient themselves towards the egg, because at this distance chemicals may not actually work. Only chemicals can help in chemical attraction in close vicinity. So what has been found by the research recently, that the sperms have got a very high power of sensing the change in the temperature. Just increase in the temperature by 0.015 degrees Celsius can be detected by the sperms. So the temperature in the fallopian tube is slightly higher than that of the vagina and uterus, and the temperature is gradually increasing if you go from the cervix up to the fallopian tube. So therefore the sperms which are attracted towards the slightly higher temperature, okay, the sperms keep moving towards the fallopian tube. That is what called as thermotaxy. Just an additional information to remember. And then now association means the sperm and egg have now come closer, and sperm binds to the egg by the reaction between fertilizing, anti-fertilizing on the surface of the sperm and fertilizing on the surface of the zona pellucida of the egg. Okay, once this happens, the sperm which reaches first to the egg will bind, and immediately what happens is, from the surface of the zona pellucida, if you take up, other fertilizing molecules are released. Suppose here one sperm has come and bind to it, other fertilizing molecules are released out from the zona pellucida. And what these do is, they attract the sperms Each molecule may be clumping around 4 sperms. Like this, the other sperms which are traveling towards the egg, they are kept away from the egg. They are not allowed to enter into the egg by a process of clumping with the help of the fertilizing molecule. This is the fertilizing molecule. Okay, so that is one way how only one sperm can enter into an egg. Then actually, before it could come in contact with the zona pellucida being recognized by the reaction between fertilizing and anti-fertilizing, before that, what actually happens is the sperm has to pass through the granulosa layer, corona radiata, and then only it will reach the zona pellucida. So a process called as acrosome reaction is important for sperm penetration. So let's see what happens here. So sperm lysins are the enzymes which are present in the acrosome, and these enzymes will help in dissolving the egg membranes so that the sperm penetrates into the egg. First to be released from the acrosome is the enzyme hyaluronidase, and that will be dissolved in the hyaluronic acid that is present in between the granulosa cells as a matrix. And then the next enzyme produced will be the coronal lysin that dissolves the corona radiata, and then the third enzyme released will be zona lysin, and that will also dissolve the zona pellucida, and then the plasma membrane of the sperm will come in contact with the egg membrane beneath the zona pellucida. So at the time of this is what called as the acrosome reaction and sperm penetration. When the leftover plasma membrane at the tip of the head after the dissolution of the acrosome, right, that plasma membrane in the form of a cone, it will be fusing with the plasma membrane of the egg, and the nucleus and mitochondria as well as the distal centriole of the sperm will enter into the egg cytoplasm, and that is what called as the penetration of the sperm into the egg. And at that time, when this penetration of sperm is taking place, the egg completes its second meiotic division, releasing the second polar body. Okay. Now, how the polyspermy is blocked, or how the other sperms are prevented from entering into the sperm. So there are so many theories which explain this, but let's take up the important one, that the cortical granules which are there in the egg, they release some enzyme, and that makes the zona pellucida highly thick fertilization membrane, right? And this highly thick fertilization membrane which is formed around the egg, so that prevents entry of any other sperm, because it cannot be dissolved by the zona lysins of any sperm. So already one sperm has entered, other sperms are prevented from entering. So polyspermy, that is entry of many sperms, is prevented. And as I have told you, the agglutination of the sperms by the fertilizing and anti-fertilizing reaction, that also keeps away the other sperms moving towards the egg. Now, formation of zygote. Fourth step, it is also called as amphimixis. Okay. Let's see how it happens. Sperm nucleus travels inside the egg cytoplasm in the direction of entry and then takes a turn and moves towards the egg nucleus and fuses with it, forming a diploid nucleus of the zygote. And this fusion is called as the amphimixis. Now, with the help of the diagrams, let us look into this process of fertilization. So, look at this. Here, two diagrams are there. In one diagram, you see that the egg is surrounded by the sperm. One is very close to it, other are entering into the outer layers of the egg. Here, this is the secondary oocyte or egg, and the sperm is making an entry. Stepwise, how the sperm penetrates, I have shown here. This is the first part of the exosomal reaction where the hyaluronic acid, the ground substance, is dissolved by hyaluronidase enzyme, and then the corona lysine released by acrosome will dissolve the corona radiata cells, and here we see that finally the zona lysins have dissolved the zona pellucida also, and sperm membrane, plasma membrane of the sperm has fused with the egg membrane, and the nucleus is making an entry, nucleus of the sperm, and this enters into the cytoplasm of the egg. And when this is happening, we see that the cortical granules which are here, right, these cortical granules, now they release the enzymes outside and change the zona pellucida into fertilization membrane, a very thick fertilization membrane. Okay, and this, in the form of a quick reaction, the change in the zona pellucida is seen in a anti-clockwise direction, and the complete zona pellucida will be converted into fertilization membrane. Okay. Now, let us take up the process of implantation. As we know that after the fertilization, the egg becomes a zygote, and that zygote undergoes cell division, forming multicellular structure or morula, and that develops into blastocyst, and that early embryonic stage blastocyst will be now attaching to the wall of the uterus, that is to the endometrium. So that process, the attachment of or embedding of the blastocyst into the endometrium of uterus, is called as implantation. Okay. Now let us go through these points first. The zygote soon divides by cleavage. Cleavage is a cell division similar to mitosis, but what happens is, after one mitosis has completed, the daughter cells do not undergo growth, they do not become larger, they remain of the same size and again divide, further forming much smaller cells. So therefore, that is the difference between cleavage and mitosis, and that is the reason why till the formation of the blastocyst, as we see here, the size of the egg remains same. It's not changing. Egg is a single cell, but it goes on dividing by the cleavage and forming a multicellular hollow blastocyst. Okay, the stage at which it gets implanted. So till then, the size of the egg is whatever the size of egg was, the same size is retained. Only the blastomers or the cells go on becoming smaller and smaller. Okay, now the zygote divides, forming 2-celled, 4-celled, 6-celled, 8-celled stage, like that. And these cells now are called as blastomeres, And the stage of 8 to 16 celled stage is known as morula, and morula further divides, increasing the number of blastomeres, and at this stage actually it enters into the uterus, and then it develops into blastocyst. So blastocyst has got one single outer layer of cells called as trophoblast, and a mass of cells at one end of the inner cavity of the blastocyst, and that mass of cells arranged like a honeycomb are called as the inner cell mass. Okay, and the fluid filled cavity is seen inside, which is blastocele. So if you look at this diagram here, this is the blastocyst, this is trophoblast layer, and this is inner cell mass, and this is the fluid filled cavity, which is called as blastocele. Okay, now okay, let's take up the next point. About the 7th day of fertilization, we see that implantation is taking place, and before that the blastocyst sheds the outer fertilization membrane which has been retained up till now. Okay, that fertilization membrane is shed out, and the blastocyst get attached to the endometrium, and soon the endometrial lining grows around the blastocyst and completely embeds it. And this embedding of the blastocyst into the endometrium is what called as implantation. Okay, and after the implantation, the blastocyst grows in size and it further undergoes differentiation, forming the embryo. Embryo grows into fetus, and after full growth of the fetus, it will be delivered out. Okay, now that's about the fertilization and implantation. Let's just have a look at the some of the pictures or diagrams here. Here what you see is the surface of the egg, human egg, and see the sperm entering here. Sperm is entering here. So that is the comparison of the size of the sperm and the egg. So sperm is very tiny in front of the egg, and you can see in this diagram the scanning electron microscope picture. It is taken by scanning electron microscope. So that is showing how the sperm is entering into the egg. Now here in this diagram we see that after the fertilization, the cleavage begins. C, that is indicating the stage of 2 cells. Then D, 4-celled stage. And you see that during the cleavage, completely the cell divides into 2. So the type of cleavage is called as here the holoblastic cleavage in humans. And then modular stage is formed, multicellular stage, that is E. And F, you see here, that represents the further growth of the embryo into the blastola stage or blastocyst. So in a section we see that it has these features: trophoblast, outer layer trophoblast, then the inner cell mass, which is made up of the stem cells and the blastocele. Okay, And this trophoblast is called so because tropha means to feed, so it helps in absorbing the nutrients from the endometrium of the uterus. And here we see that the blastocyst has implanted into the endometrium. So with this, we'll conclude this part of the study. We'll meet you in the next session.